You don't have to read all the particulars of these slides. So in 2013, the ex-wife of one of the Google founders set up a company as a hobby, as you do, when you've got spare billions. And she said, I'm going to transform medicine through genetics. And so she set up a company called 23andMe, which in 2013 was shut down by the FDA. What this company has done is they've genotyped about 900,000 people in the United States. So there's consumer demand for this stuff. But she was spruiking very limited clinical evidence to say, oh yes, you've got this gene variant. There was one study of 2.5 people that showed you will have schizophrenia and die next year. And so they were producing reports that were generating acute adjustment reactions, catastrophic reactions. People were attempting suicide in the United States. Obviously, the regulators have to protect the vulnerable from snake oil, be it genomic snake oil or fish snake oil or any other kind of oil. And so they shut this company down despite it being one of the most powerful startups in America with its backers. And this really brought the tech industry into line with healthcare. So very different from an app that is crappy. Healthcare, society must protect the vulnerable. And so all of a sudden the San Francisco set discovered the difficulties of getting startups going in healthcare because you need an evidentiary threshold first. So this is really how not to proceed in this space. And there was a lot of fear after this about other companies going the same way. And after this company was shut down, I noticed more and more people were asking my advice from industry. And only one company, as I mentioned before, said, how do we do it responsibly from the start? Because I think they realise that if they try and spruik snake oil, when there's serious clinical implications, they will and should be shut down. So it's good to know the regulators are still there. But like any kind of disruptive change, they're still figuring out what to do. Now, long story short, in just a cartoon form, genetically guided prescribing or precision medicine, also often called personalised medicine, is really the current practice when we prescribe a therapy, principally biological ones, but so we prescribe a pill, is there'll be a mix of people from the population who get different outcomes. And in part, this is influenced by our genomic variability. So you'll get a mixture of people who, some get a happy result, some get an unhappy result. The hope with some genetic guidance is, and PGX is shorthand for genetics. So RX is treatment, DX is diagnosis, you know that. No PGX. That can be a shorthand. It's worth memorising that one. Your notes will look more competent if nothing else. PGX just means genetically guided prescribing. Whether we can actually shift it so that the odds of having an adverse drug reaction like Stevens-Johnson syndrome or psychotropic-induced suicidality or clozapine-induced neutropenia are reduced, and increase the number of people who get a good outcome. The holy grail is whether we can separate out whether psychosocial stresses drive a presentation or not. I'm not sure that we'll get threshold in the next decade for that. So the hope is whether we can screen out some of the trial and error. Basically, this is all that happens, is you get people from that mixed population. You get a sample, which can be a saliva sample, be it a buccal brush from the inside of the cheek, be it a spit sample. Spit samples are a bit better because they're more accurate when they do the genotyping, or a venous blood sample. And then they put it in a gene chip. So these are kind of mass-produced things which enable you to read certain gene variants. They're affordable. Labs pay about $25, $30 for one chip. They should stick it in their machine. And then whether that'll give you information that actually helps to clinically stratify people. But again, the key is, what's the evidence? And we'll go through this later. Put another way, often with the process of trial and error prescribing, there's a delay to care. So time is money. So I think for most of us in psychiatry, we're accustomed to a prescribing odyssey often. Sometimes we get lucky and the first thing at the first dose does the job and we feel like a hero and we're happy for the patient. But often it can be months, sometimes years. And one of the things that got me interested in this space was, in my first year as a psychiatrist, seeing somebody who had seen their GP for 2 years was on 225 milligrams of venlafaxine, had a persisting depression, quit their job, attempted suicide, lost their wife. And because I had the medico-legal license to say, okay, we'll go above 225, we went to 450 and they remitted. There was no childhood trauma. There was no other psychosocial adjustment stressor perpetuating their depression. There was no substance abuse. And I thought, gosh, why did they have to wait 2 years just to see me? So that for sociocultural reasons, I was a designated higher witch doctor that could just go beyond the MIMS level. There must be an empirical way, hopefully. So again, the hope is whether we can actually reduce trial and error prescribing, reduce time to response remission, and in that time saving, save ourselves as clinicians time, more importantly, the patient and their loved ones time, and important at another level, save society time, which equals cost. So if we can actually save cost, speed recovery, and free up clinician time, it's almost like a holy trinity of efficiencies. So you can see why there is a lot of hope, but again, needing to be backed up by evidence. Now, this is in your handout. It's a slide from a German-led trial where they followed a whole lot of people with depression, did rating scales, and looked at genetic variants to see whether you can see an association between gene variants and different outcomes. I think it's one of my favourite pictures from the empirical literature. So often the empirical literature, overwhelmed with the complexity of complex systems like the human mind and a need to come up with an implementable nosology, cartoonifies reality into simplicity. This is what we see as clinicians. Somebody turns up with depression, we put them on a pill, we decide that a pill's the approach for their presentation, and you get this multiplicity of outcomes over a 12-week period in this instance. And this to me is what we're faced with every month as clinicians. Some people will do beautifully, you put them on a pill and it's like, holy cow, that's great. Wow, they've remitted by 3 weeks. I told them that's what the PI said and it happened. Hooray. But then you've got other people that get worse. Some in between, they sort of respond but don't remit, they stay miserable, don't do all the psychosocial things to perpetuate their recovery and invariably relapse later. So the clinical reality is whether, this is the clinical problem, but the question mark is whether we can pick out who's going to be like this and really push a biological approach more, or whether we can figure out why this person's not getting better. Is it non-biological factors underpinning their perpetual depression, or is it in fact they've got unusual metabolism and need a different type or dose of medication? This is a meta-analysis looking at from a single trial of antidepressants, SSRIs, mirtazapine remission. So when I started going into depression research, I realised that there were all these fake outcome measures, like percentage reduction in the depression scale, or half better, AK response. Whereas what matters in clinical reality and in terms of functional recovery is remission. Essentially, virtually all of the symptoms go away. So when you look at remission as a real-world translatable outcome for efficacy in depression studies, and this talk's going to be mainly around depression and antidepressants, so that's what I work in, it's about 40% remit from a single trial. Now we know as clinicians, if we coach them, we keep going, you do several trials, very much like shown in the STAR-D data set, and you'll get incremental improvements for some, assuming there's an impart or principal biochemical underpinning to the depressive presentation. But it's pretty low rate, and when you think 350 million people globally have major depression according to the CDC and World Health Organization, when you think that 10.8% of Americans aged 12 and over are currently taking an antidepressant, there's a big public health sort of issue and question mark whether we can get improved outcomes. So there are real problems to which the technology is being applied.